Okay. Sorry. After the interruption, I also had an issue with the network. So I think we are able to explain the behavior of phospholipids and how a phospholipid bilayer is formed and why we have a membrane that is self-healing and what are the contributors of a membrane being greedy in nature and how being greedy help in ensuring the activity and the function of a cell membrane. The next thing we talked about, and this was mentioned in your lecture material, the asymmetrical nature of lipid bilayer. Are you with me?
Yeah, with you, sir.
Very good.
Yeah,
we are with you, sir.
Thank you. Sorry, I was explaining something about liposome, right?
yes, sir. Yes, sir.
Yes, I was explaining something about liposome, and I said a liposome, apart from this is the structure that is formed automatically for a cell membrane. You have the bilayer, lipid bilayer, and then inside, like here, you have the cell component. But I said that this structure can also be artificially formed so that it can ease drug delivery. And we mentioned why we encapsulate drugs. Apart from that, we also mentioned that some drugs find it difficult to reach the target organs. Most especially, I don't know if you may be familiar, there is what we call pharmacokinetics and pharmacodynamics. So specifically in pharmacokinetics, we talk about the interaction of the drug with the system. How does the system act on the drug? This is purely pharmacokinetics. There are some drugs that before they reach their target organ, they got metabolized. Or maybe the physical property of the drug, the solubility, may not allow the drug to reach the target organ. And I think that is one of the reasons why you will have the situation that some drugs are administered in a different route. You can have oral, some you don't even have oral. They must be IV. Some they are intraperitoneal. Some you inhale them. Why? Because of the nature of the chemical and how does the chemical interact with your body. There are some drugs that you have IV, but you don't have oral. Simply because when you have them oral, before they reach the targets where they're supposed to act, maybe they'll get metabolized or something will happen to it. Or the physical property will not even allow it to be absorbed right from the intestine. So all things like that are some of the things. But in personalized medicine, where you have a particular situation, let's say a cancer of a particular organ, let's say a lung cancer, you have a drug that you want that drug to specifically target the cancerous cells, because even in the cancer, in the lung, not all the cells are cancerous. For example, there are some portions within the lung that these cells are already affected. They are cancerous. They are out of control. How do you have a drug that will target those particular cells that are cancerous? So this structure is used in medicine. You create artificial liposome, and this liposome will contain some. Are you with me, class?
Yes, sir.
Will contain okay, will contain some proteins, recognition proteins. We'll talk about glycoproteins, we'll talk about glycolipid and how they are important in identifying or facilitating interaction between cells. And like here in liposome also, these particular glycoproteins are added in the liposome so that they can specifically interact with those cancerous cells. Because in cancer cells, in all the cancer cells, distinctly, in lung cancer, for example, there are some proteins, some glycoproteins that have been expressed. That's what we call cancer-specific immunoglobulins. So each cancer will have their own specific immunoglobulins. Even within the same cancer, you can extract lung cancer cells, check their membrane, you can find out some immunoglobulins, some glycoproteins that are only specific to those cancerous cells. If you are to extract the cell of the lung, the same lung, where there is no cancer and where there is a cancer, and you compare the composition. Rahab, please. So, when you extract, you will discover that there are these immunoglobulins that are only specific to cancerous cells. So you may design your liposome to target those immunoglobulins specific to the lung cancer cells. So when they get there, they can easily interact with those glycoproteins, and that interaction can facilitate their own entrance or releasing of the content of the liposome. So you may decide to put those drugs inside the liposome. The liposome contains these glycoproteins. So once ingested, they go to the lungs. Even when they get to the lungs, they will not interact with any type of the cells, but those specific cancerous cells that contain these glycoproteins. So they interact with them, and that mediates the release of the content of the liposomes into the cancerous cells, and hence the killing of the cancerous cells. So it is a very important career mediator in personalized medicine. I want to believe you come across, even though we have not reached that advanced stage, but as time goes and you are into the area, you will definitely come across this personalized treatment. So before we move, we talk about the asymmetry of phospholipid. Sorry, asymmetry of plasma membrane lipids. Okay. This was mentioned in your listing. Some membrane, for example, red blood cells. Remember, we talk about bilayer, right? There is an upper layer, for example, here. There is an inner layer, or an outer layer and an inner layer. And we made mention of a different phospholipid: phosphatidyl serine, phosphatidyl choline, phosphatidyl ethanolamine, etc. And for every cell, the compositions of these phospholipids are not the same. Most especially for a particular lipid also. When you look at the two monolayers, the upper layer and the down layer, they have a completely different composition of phospholipid. Now we give an example of red blood cells where the outer layer contains a phosphatidyl choline and sphingomyelin. So mostly you would only see phosphatidylcholine and sphingomyelin in the outer layer of red blood cells, while the inner layer contains phosphatidylserine and phosphatidylethanolamine. This is basically what you will see. Only in some certain situation, now, in addition to that, there are glycoproteins that are glycolipids usually found in the outer layer, because like I just said, the glycoproteins and the glycolipids are mostly involved in recognition, serving as receptors and the rest. So mostly they are found in the outer layer so that they can interact with the outer environment or extracellular environment. Now, why this asymmetry of lipid bilayer? It is significant in understanding. For example, let's give a typical example. We talked about the lifespan of a red blood cell after 10 to 12 days. When a red blood cell reaches 12 days, it reaches its own lifespan. Now, there will be what we call apoptosis, programmed cell death. So that cells will die after 12 days. How do you know that this and this and these cells are dead? Yes, of course, a lot of them that reach that 12 days will die after 12 days. So how do you know specifically these and these are dead? Class, are we together?
Yes, sir.
You have an idea. Now, how does the body identify cells that are dead? For example, the red blood cells, after reaching the 12 days of their lifespan.
Could we say that in some parts of our body, like the epithelium, that's our skin, we find these dark spots sometimes that when we ask questions, we're told that they signify dead cells?
Yes, that's a good answer to skin cells. Yes, skin cells, when the cells die, it can be dark, and because it is the skin and the outer covering, over time you can even brush it out and remove or something like that. But when we are talking about cells that are inside, for example, red blood cells. Any idea?
Hello, sir.
Yes, I can hear you. Or Samuel, right? Did I pronounce your name right?
Yes, sir.
Okay, go ahead.
Okay. For red blood cells, can I say that it can lead to anemia?
Sorry?
Can I say that for the red blood cells that it can lead to anemia?
That is when you have when the red blood cells is no longer doing what it's supposed to do, it can lead to anemia. But this is completely something else, different from what we were discussing.
Okay.
Like when the red blood cells are dead, how does the body know that these cells are dead?
Can fatigue be a sign?
Sorry?
Fatigue.
Fatigue. You see, all these things are more of physiological. I'm talking about inside the cell, physiologically. How could the body machineries know that this cell is dead? And you need to be clear. You need to go out of, need to be removed completely. Okay. I thought you have actually read. Helen, can hear you well. Can you speak louder?
I said it goes out by a process of Phagocytosis.
Yes, Helen. Phagocytosis, a process where all these unwanted substances are removed by white blood cells. So the question is, how the white blood cells identify that this and this are dead, therefore they need to be phagocytosized. Okay, now the explanation here is we just talked about the composition of phospholipid in the plasma membrane. And we made mention that phospholipid like phosphatidylcholine, sphingomyelin form mostly the phospholipid that are found in the outer layer of the membrane, the monolayer. Is that clear? Are we together?
Yes, we are.
Yes.
Yes.
While the inner layer, for example, here, let's say, let me look at this. Let me give an example of this liposome. We have this as the outer layer that interact with the extracellular fluid, and this as the inner layer that interact with the what? The intracellular fluid. So the composition of this phospholipid mostly comprises of phosphatidylcholine and sphingomyelin, while here mostly contain phosphatidyl ethanolamine and phosphatidylserine. Osamui, can you mute yourself, please? So we have this composition. So in no reading, you will see phosphatidylserine or ethanolamine in the outer layer. By the time you see phosphatidyl ethanolamine or phosphatidylserine in the outer layer here, it signifies something. It is a signal. So in red blood cells, we say presence of phosphatidylserine in the outer layer indicate that this cell is dead. Is a signal that this cell is dead. Are we together? So I don't know, but we don't need to talk about this, about the movement of the phospholipid, because the fact that these phospholipid are packed here, some are in the outer layer, some are in the inner layer, they still also move. They can undergo translational movement. You can have a phospholipid from point A moving to point B. For example, let's say you have this one here at point A, and you have this one here at point B. You can have a translational movement. This one can move to this place, while this one can move to this place. Likewise, you can have A also moving to here. For example, let's say this one is C. So this one can shift down up here, while this one can shift down here. We call this flip-flopping. And it is an enzyme called flipase that helps in this flip-flopping movement. So in a situation once a cell is dead, in the case of red blood cells, you have a phosphatidylserine that initially form the composition of the inner membrane will now move to the outer membrane. So movement, seeing a phosphatidylserine in the outer membrane or monolayer shows that this cell is dead. Therefore, the macrophages can come and phagocytosize, take up this for killing. Is that clear? Do we make any sense here? Do we make any sense, class?
Sir, please,
Yes.
if I heard you correctly, did you say that I really might not be able to pronounce what you pronounced? Maybe phosphatidylserine something. Did you say
Phosphatidyl, yes.
okay? Did you say it was on the outer membrane and then we now found it in the inner membrane, or did it go from inside outside? I want to understand the placement. Did it come from outside
It is
into the
from,
inner membrane?
So listen to me. I said each composition, each cells have its own composition of this phospholipid. Remember, I told you a different type of phospholipid, right? And for example, I said there is phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylinositol, etc. Right? Are we together?
Yes, sir.
Yes. So, in asymmetrical nature of phospholipid, we said some specific cells, for example, red blood cells. The composition of phospholipid in the outer and the inner monolayer differs. In red blood cells, we have phosphatidylcholine and to some extent sphingomyelin form the phospholipid in the outer monolayer. While the inner monolayer comprised majorly of what? The phosphatidylethanolamine and phosphatidylcholine for a living cell. Once a red blood cell reaches the lifespan, it is dead. The phosphatidylserine that normally is supposed to be in the inner monolayer can now translocate to the outer layer so that you will have phosphatidylserine in the outer layer. This movement and the presence of phosphatidylserine in the outer layer is a signal that this cell is dead. Therefore, immune cells responsible in clearing unwanted substance in the body will now take it up and then digest it. Is that clear now?
Sir.
Is that clear, Sharon?
Sir.
Go ahead.
Thank you, sir.
Okay, you're okay now.
Yes, sir. Yes, sir.
Do we have any question? Sorry, I can't remember this one. Kwatongo. Did I pronounce your name right?
Yes, sir.
Are you clear? Did you get what we are saying?
Yes, sir.
Good. Okay, now let's make a progress.
Okay, sir. Thank you.
Okay. Remember, we are still talking about the lipids found in membrane. So we just talked about phospholipid and how these phospholipids actually form the major components and how are they oriented in the phospholipid and how do they even determine the bilayer? How do they determine the self-healing property, the asymmetric property? And we talk about the fluidity. In addition to that, we have cholesterol. Cholesterol are also found in a cell membrane. They are also amphipathic in nature. In addition to that, they improve the stability of the cells. I actually wanted to put the structure of cholesterol so that you can see the polar region and the non-polar region. It has a kind of hydrophobic, you can see the structure here from the image here. You can see some hydrophobic compounds that form part of the membrane, both the inner and the outer. So it has an OH group. That OH group is what contributes to the solubility aspect of the cholesterol. And that's how in increasing the polarity of the polar region. And in addition, it forms like a kink bond trying to prevent the compaction. We can see the polar region here around we can see the aromatic rings within the fatty acid chain. And we said if the fatty acid chain are saturated, They can easily compact together. But now we have a structure that is not straight like the rest. Therefore, it will prevent the compaction. Therefore, the membrane will still remain fluidy in nature. So it's increase the fluidity of the membrane. Is that clear? Is that clear?
Yes, sir.
Good. So the last one, we have glycolipids. And glycolipids, they are just like a phospholipid, but the difference is that in phospholipid, we say the backbone is glycerol, whereas in glycolipid, mostly the backbone is pangosine with just a fatty acid attached to it. And the last where this choline, ethanolamine, and serine are added, you have a sugar added instead of these compounds. It can be a one monosaccharide, it can be two sugar moiety, and the rest. When one is added, it is called cerebroside, which of course we will discuss when we are discussing chemistry of lipids. When more than that is added, we have what we call ganglioside. Is that clear? We also have proteins.
Yes, sir.
So now we just finished talking about the lipid found in the membrane. The next one is protein. There are also proteins that are found in the membrane. They are of two types. We have peripheral membrane proteins and we have integral membrane proteins. And altogether, they play a very important role either as receptors, either as enzymes, transporters that allow the movement of substance in and out of the cells. There can be channels that allow the movement of electrolytes like potassium, calcium, sodium, etc. They can also serve as recognition molecules like glycoproteins, just like we mentioned when we are trying to discuss the liposome specifically interacting with immunoglobulins in the cancer of cells. Is that clear? So it can be peripheral or integral. It is called peripheral when the membrane, when the protein is only found on the surface of the monolayer. For example, we can see peripheral membrane protein here. It's a protein that is just attached to the periphery, either it's inside or outside. Is that clear? We can see peripheral membrane protein here. And we can see integral, the one that started from the outer down to the inner monolayer. Is that clear? Class, are we together?
Yes, sir.
So this is basically peripheral membrane protein and the integral membrane protein. And we made mention what are they used for. Well, usually they can be receptors that combine to something and serve as recognition molecule. They can be an enzyme, catalysis. They can be transporters that help in the movement of substance in and out of the cells. They can be a recognition that determine what it is before it gets into the cell. That brought us to the last aspect of study session 2. Before then, we talk about glycoproteins. Remember, we said what glycoproteins they are used for, right? They are used for what? For identification. So on the surface of the outer monolayer of the membrane, we have glycoproteins. They are called recognition molecules. We can see glycoproteins here. Some of them are here, they contain. So most of these glycoproteins, you can see a protein here, and then what we have here is carbohydrate attached to it. And here we have a fatty acid, and we also have a carbohydrate attached. This is what we call glycolipid. One of the functions of these glycoproteins is in the identification of blood grouping system. I am very much sure, even though you are not for some of you that are already practicing nurses, you must have known at least about blood group, right?
Yes, sir.